Hello. Today we will be talking about molecular testing in the blood bank lab. We'll start with some basics. What is DNA? It is a polynucleotide formed from covalently linked deoxyribonucleotide units. It serves as the store of hereditary information within the cell and the carrier of this information from generation to generation. DNA is a double-stranded molecule composed of nucleotide subunits. Those subunits contain a phosphate group, a deoxyribose sugar, or in RNA it would be a ribose sugar, and nitrogenous bases, which are either purine or pyrimidine. The purine bases are adenine and guanine, and the pyrimidine bases are thymine and cytosine. A pairs with T and G pairs with C. The sugar and phosphates make up the backbone of DNA. Coding strand runs 5' to 3', and that is important when you're designing primers for PCR. The anti-parallel pairing of AT and CG creates the double-stranded helix. The genome is the complete set of information in an organism's DNA, and it carries all the information for all the proteins the organism will ever synthesize. Each genome contains a ton of information. A typical human cell contains 2 meters of DNA, yet the nucleus of a human cell is only about 6 micrometers in diameter. So how does all that DNA fit into our cells? The DNA is wound around proteins called histones. This forms nucleosomes. The dense, compact DNA protein structure is called chromatin, and when the cell is not dividing, the chromatin condenses even further into chromosomes. So this is a really nice image that shows DNA as it is, adding the histones, then it makes that bead on a string formation. It winds even tighter into these fibers. It forms scaffolding proteins, and then even thicker into chromosomes. A gene is a region of DNA that controls a discrete hereditary characteristic. A gene is composed of 2 regions, exons, the coding regions, and introns, the intervening regions that do not code for a protein, and they are removed when mRNA is produced. RNA is transcribed into single-stranded mRNA. During translation, the ribosome will read the messenger RNA 3 bases at a time. Those 3 bases are a codon, and they correspond to an amino acid that will be translated. Now we're getting into the testing part. PCR, polymerase chain reaction, was invented by Kary Mullis in 1983. He was working for a company called Cedis. He ended up winning the Nobel Prize for this invention, and Cedis gave him $10,000. Cedis turned around and sold PCR to Roche for $300 million. There are 3 main parts of PCR, which we will go over in the next few slides. So PCR is a tool to basically make a lot of copies of the DNA that you're looking at. So you start with primers. So you want to design your primers to flank the target region that you're interested in looking at. To start the PCR, you want to add your DNA template from your patient or donor that will contain the target somewhere in the DNA. You're going to add your primers, free nucleotides, or dNTPs, and then DNA polymerase. This will be placed in a PCR instrument, which we call a thermocycler, and it will increase and decrease the temperature, which will help with these steps. First, it will be denatured, so your double-stranded DNA will become single-stranded DNA. And then the next step is annealing. This is when the primers will anneal to your DNA. So once your primers anneal to your target DNA, the temperature will be increased to optimize DNA polymerase action. It will catalyze the addition of nucleotides to synthesize new strands of DNA, starting from the primers. And this process will repeat over and over. So in cycle 1, you started with 1 copy and ended up with 2 copies. In cycle 2, you started with 2 copies and you'll end up with 4, and so on and so on. And this is just another way to visualize it. So for example, if you go down to cycle 10, you'll have 512 copies. And then once you go down to 30, you'll have a lot of copies. And normally we do 30 cycles during a PCR process. And if you'd like to see it in action, if you just click on this link, it'll take you over to the Bio-Rad website that will explain it in detail. So molecular is a growing field. There is an ASCP exam only for molecular biology, which you can sit for after a year of experience. We use it for a lot of different diagnostic testing. You can use it for cancer screening, genetic counseling, newborn screening. You can use it for cytogenetics. Pharmacogenomics is a growing field that can help doctors know if a drug will help you or hurt you. And then we also use molecular testing in HLA and transplant and platelet testing. And we use RBC and HPA genotyping and blood banking. And then, of course, Forensics uses DNA testing as well. So how do we use DNA testing in the blood bank? Monoclonal antibody drugs are growing. Specifically, the ones that we deal with in blood banking are anti-CD38, which is also known as DARA, daratumumab, or Darzalex. And then anti-CD47 is a new one. They cause a lot of problems because CD38 is also found on RBCs. So the patient is getting this drug, and it's basically making everything look positive, so you can't really do antibody rule-outs. Check out this link. It is very useful, has a lot of good information in it to tell you all about the testing that we do to get around the interference of these drugs. It's also very helpful whenever we don't have any known antisera for that certain antigen. So in the cases of Dombrach, Holly, Joseph, some of the RH antigens, and U, we also use it for DAT positive or multiply transfused patients. We can't get a phenotype because the patient's just been transfused, so we can do a genotype. We also do mass donor screening to look for special antigen units so that we can match up donor units with patient needs. It's also useful for distinguishing between weak D and partial D phenotypes. And we can use it to resolve allo versus auto antibodies. We also use molecular typing for platelet genotyping, which will be discussed in the platelet lecture, and also in HLA typing, which will be discussed in the HLA lecture. So this is a little summary of what events can cause certain phenotypes. This is located in the technical manual. Some changes in antigens are caused by SNPs, which we'll talk about in the next slide. Some can be affected by silencing mutations. And then we have more complex groups like ABO and RH and MNS that have a lot of factors kind of compounding on each other. So SNPs are single nucleotide polymorphism. To be called a SNP, it has to occur in 1% or more of the population. And a lot of RBC antigens are caused by SNPs. They are naturally occurring. They occur every 100 to 300 bases. It is just a change of a single base pair in the triplet codon. They can be silent, in which case it would not change the amino acid that it codes for. It could be a missense SNP, which does change the amino acid, and then it could be nonsense, which results in a stop codon. SNPs can be naturally occurring, passed down from your parents, or they can be caused by errors during replication, or they can be caused by DNA damage. And like we said on the last slide, they can cause altered proteins, or they can just be silent SNPs. There's also larger variants caused by structural variations, and there's a lot of different things that can happen. You can have inversions, you can have translocations where part of one chromosome is actually inserted into a different chromosome. You can have deletions, duplications, and insertions. Once we've performed the hybridization, we will acquire the beads on the Luminex, which is a flow cytometer. And then once we have the raw data, we'll analyze that. So this is kind of a breakdown of what happens. We have these Luminex beads, which have a capture molecule, which in this case is an oligonucleotide. If the complementary sequence is present, it will bind to that oligo, and during PCR, biotin is added into the sequences. So when we add the SAPE is our reporter molecule, it will bind to the biotin if that complementary sequence is there. So how the Luminex works in general, all the assays are bead-based. The beads have a mix of red fluorescence. So you can imagine a map, and based on the fluorescence of the red, it's going to land into one of these buckets. So for example, this bucket may be for JKA, this bucket may be for JKB, and you want to have beads in each bucket, because if you don't have any beads, then there's something wrong with your assay. You lost the beads somewhere in the process. But the green laser is what the SAPE is going to give off. So in the JKB bucket, if you see your beads and then you also have green fluorescence, then that patient or donor will be positive for JKB. So you'll always have red fluorescence in all your buckets, but the green fluorescence is what tells you if that SNP is present or not. And this is a really nice assay. We can test 96 samples at a time. This is an example of a genotype report for a batch. So it will tell you over here the DIN that was tested, or the donor identification number. And then the top tells you all the alleles that are assayed in this test. So all of this, each donor or patient is one well, and it's testing all this information. So it'll give you your genotype for each of your samples here. And then we also have a phenotype result. So we discussed in the genetics lecture, when you're doing DNA genotyping, you can only give a predicted phenotype because you're not actually testing the red cells. So you always want to say predicted phenotype, because there's always things that can happen in between DNA to protein or DNA to antigen. So, and we do find that sometimes our DNA type will say they should be positive for big C or big K, but then serological testing will tell us that they're negative. And then we have to investigate what's going on. Another note that is really nice about these reports, it tells you what polymorphism it's looking at here, and then what polymorphism it actually is detecting in the assay. So the first nucleotide is usually the reference nucleotide. So normally you'll have an A here at this location, but a change to a G can cause the phenotype. So you can see this patient only has 2 A's, so they have a normal reference allele. And the bioarray test is a little bit different. We'll talk about that on the next few slides, but that is something that's really nice about this assay, is it actually tells you what nucleotides are detected in the assay. So the other assay that's on the market for RBC genotyping is bioarray, or HEA, which stands for human erythrocyte antigen. It is not an assay that we love. We were doing a ton of testing with this assay, and we got a ton of failures. It works kind of the same in that the beads have an oligo sequence that it's looking for. The difference is they bind these beads to a chip. So you see there's a 96 well chip that each well has a chip on it, kind of like this, and the beads are bound to it. And beads don't do as well when they're bound to a surface as they do when they're free-flowing and liquid, as they are in the ID-Core assay. You can also see there's a lot more steps in this assay, so it was very time-consuming, but it was the same type of methodology. You do your PCR, and then you bind your amplified DNA to your beads, in this case, chip, and if it was the complementary sequence was present, it would bind to the bead, and if it was not, then it would not. And then you take a picture with a fluorescent microscope, and you get an image that looks like this, which we cannot decode. It's so teeny tiny. There's a software that will basically know what each SNP is at each location, and then it will give you a phenotype. So going more into the analysis of bioarray, you'd have the intensity for each bead. So the SNP would be down here, and most of the SNPs only have 2. For example, like the Duffys, you have Duffy A and Duffy B. So they have a cutoff for each to call it positive or negative, because some just have background fluorescence. So then you might, in this case, have a negative Duffy A and a positive Duffy B. So this is the part that we did not like about this report. This is their genotype report. You can see it tells you what polymorphism it's looking at, but it doesn't tell you what nucleotide it actually detected in the assay. So it'll tell you it either saw the A or B, but it doesn't tell you which one is which. So by looking at this, we don't know, is A the C or is A the T? So this report was very complicated. We elected not to share this with hospitals because we would be answering questions all day long. Even for genetic people, this report was confusing. So we liked the ID-Core report much, much better. So this was the report that we gave to hospitals for patient testing. It is the phenotype report, so predicted phenotype, of course, and it would tell you the group, the antigen, and then the predicted phenotype, positive or negative. These are the antigens that bioarray detected. It did have a nice variation. It had some of the more rare blood group systems, such as cyana. It is not on the ID-Core assay. So the predicted phenotype calls from this assay are based on these polymorphisms. So you can convert what polymorphism is present to the ISBT phenotype or genotype. Another assay that we use in blood banking is real-time PCR. Real-time PCR is useful in that conventional PCR just tells us what, and real-time PCR will tell us what and how much, because it's actually taking readings during the amplification process. So you can see how much is present. So the assays that we use for real-time PCR, we don't actually use it for its quantitative nature. We use it because it's a very fast assay. The downside is you can only test one patient or donor at a time, and each well is only positive for 1 or 2 SNPs, as opposed to the multiplex PCRs that we're doing for ID-Core and BioArray. So, for example, each well would be a different SNP. So you might have well 1 is JKA, well 2 is JKB. So over here, each color would be your different well. Down here will be your cycle, and then over here will be your fluorescence. And then you have a cutoff value. So once it goes past the cutoff value, you can determine that that well is positive for that SNP. This can also be visualized if you gel that plate. Each well you could visualize this way. So one of these is A, we'll call A, B, C, D, and you have an expectation of where that SNP would be. Usually we would only see either it's there or it's not. And then each well also has an internal control. So we would expect to see an internal control up here, and then a SNP whatever size we would expect that to be. Real-time PCR can be kind of a hassle to analyze because some of these assays are a 96 well plate, one patient, and you have to look at your positive locations and then kind of add things up to determine what the phenotype or genotype is. So it can be quite complicated. So like I said, it is very nice because it's very fast. You can get a full genotype in 90 minutes, but it is very expensive because you can only do one sample at a time. So we're also bringing in next generation sequencing into my lab. It's very nice. You can test 384 donors at a time. So the more you multiplex, the more cost effective it is. This methodology is really nice. You get a ton of information in one shot. The downside is because you're testing so many at one time, it can take a while to find that many samples to test. And then the actual sequencing process is about 24 hours. But it is highly sensitive, very accurate, and you're getting a ton of information. And the way you can pool so many samples together is you amplify all your targets that you're interested in, and then you're tagging that amplified DNA with a certain barcode. So each sample has a different barcode sequence. So after you barcode them and pull them all together, they run on the flow cell, and then you can tell according to the starting barcode what sample that belongs to. So there's lots of advantages to this. It's a ton of data. It's very cheap because you can pull so many together. The disadvantages is the interpretation can be complex. They are developing some software where you can just kind of throw your data in there and it can spit out a predicted phenotype for you, but those are still kind of in development. So it does take some skill, and obviously because you're running so many samples at a time, you want to make sure that you're doing it well, or it can be very expensive to lose all of that data for one run. So William Lane developed this software called BloodTyper. He used the 100 Genomes Project data to, he by hand analyzed the data to come up with a predicted phenotype. Then he developed this software so that he could put the data in and it would spit out the phenotype for him. So pretty much you're doing your sequencing, you get all this data, you upload it to the software, and then it spits out this very detailed report of all the predicted phenotype. And that is genotyping in a nutshell. Please let me know if you have any questions.